Okay, so now we're going to discuss the prefixes and the suffixes in DNA synthesis. You can skip this, however, I strongly recommend that you know them before we start the lecture. It's going to make things so much easier. So, for you that don't know what a prefix and a suffix is, the prefix is simply something that we put in front of the word and it changes its meaning. The suffix usually comes at the end of the word. Basically, if you know the prefix and the suffix, you'll have a general idea of what the thing is even before we discuss it. So let's start with the first prefix we're going to see, and this is the prefix called DE. Okay, so what does DE mean? DE means lacking, to lack, right? Damn, I have very bad handwriting. So to lack, or it means without, right? So without or lacking what? For instance, decalcification, right? So lacking what? Calcification, calcium. So it's lacking calcium. Dehydration, for instance. Right? Dehydration. So lacking, what's it lacking? It's lacking hydration, which is lacking water. Depigmentation. Depigmentation. Like, you know, some diseases like vitiligo, right? So lacking what? What's it lacking? It's lacking pigmentation. Deamination. Deamination. D, lacking what? It's lacking an amino group. Okay, hydro. So again, and we talked about this, hydro simply means water. Okay, so things like hydrolysis, for instance, hydrolysis. Hydro means water, lysis means, well, to break something, right? So that is it, hydrolysis. Hydrophobic, for instance, hydrophobic, right? Hydro means water, phobic comes from the word phobia, which simply means fear, right? Hydrophilic, Philic means loving, hydro means water, so it loves water. Hydrotherapy, for instance. Hydrotherapy, right? So what's hydro-water therapy? So basically therapy or treatment using water. Number 3, lysis. Lysis, so stuff like hydrolysis, and we talked about this, right? Hydrolysis, so water breaking or breaking using water. Glycolysis, and by the way, this is no longer a prefix, it's a suffix because it comes at the end of the word, right? So glycolysis simply means breaking down the sugar, right? So where do we see the suffix is? And this usually see it in enzymes, so something like polymerase, right? Or kinase, or ligase. Again, this is going to be a suffix. Here's is, right? Basically, always when you see something like this, know it has to do something with an enzyme. And the last one is the O's, right? So basically, when you see this, you know that's going to be related to sugars, right? So ribose, for instance. And by the way, again, this is going to be a suffix, ribose. Glucose, right? Deoxyribose, pentose. Okay? So O's, you know directly that this is going to be a sugar. So let's recap really quickly. D means to lack or without, depigmentation, dehydration, etc. Hydro means water. Lysis means to break. Is a suffix. Usually it's going to be used something related to an enzyme, right? Now, don't worry about this page. It's simply the core molecular structures that you must know. You don't have to know the chemical structure to it. Just know the formula for it. For instance, when we say hydroxyl, just know that this is hydroxyl, right? When we say methyl, it's called CH3. Amino, for instance, is NH2. Phosphate, we don't even need to know carbonyl, but I don't think we're going to talk about it. Phosphate, PO4, right? Pyrophosphate, you know what? Just pyrophosphate, let's do it like this. This is pyrophosphate. Okay, carboxyl would be something like this, right? COOH, and that's all. Now, you don't have to memorize these, just get a general idea about it. Now, without further ado, let us start our DNA synthesis lecture. Today's subject is going to be about DNA synthesis, and we're going to insert RNA synthesis also with it. Why? Because basically, on a molecular level, they're extremely similar, okay? So, the way we're going to do this is that we are going to take a double-stranded DNA. We're going to start unwinding it. We're going to break it down and keep breaking it down until we get to the core bases that make the DNA. Okay? So, let's start. Now, this is going to be a double-stranded DNA. Okay, here they have, of course, this is a 3' end, a 5' end. Don't worry about it. and the other one also has a 5' end and something called a 3' end, right? So let's take this double-stranded DNA and let's break it down to the core, right? So first of all, let's unwind it. Okay, great, we unwind it, unwind, right? And you have something now like this. Again, it has a 3' end and a 5' end. Let's read it out, make it into, okay. So, first of all, sticking out of it, like this, are things called nitrogen bases. All right, nitrogen bases. Don't worry about it. Don't worry about it. I'm going to talk about this. Okay. Anyway, suppose that you know we took this. Of course, this is called now a single-stranded DNA. In textbooks, in medical literature, we call it a DNA molecule. Okay, it's called a DNA molecule. Now, suppose that we take, let's say, a strip, of course, nitrogen bases. We've got millions and millions of them. Suppose we take, let's say, 50 of them, right? And so now you have a, well, it's no longer called, I'm not going to draw 50, but you know, you get the idea. Each line here is going to be representing a nitrogen base, okay? And since they're more than 20 to 25, since they're more than 20 to 25, we call this polynucleotide, okay? Now, suppose out of these, let's say there are going to be 50 nitrogen bases here. Let's say, suppose we take, I don't know, like 15 of them, right? So now we have only this small strip. It has 50 nitrogen bases, and now we call it an oligonucleotide. Listen, basically, I've got to tell you what a nucleotide is first. Stay with me, okay? So let's suppose that here there's a nitrogen base. Now, let's suppose we took only one nitrogen base and we put it under a like electronic microscope or whatever. This is what we are going to see, right? You're going to see that this is called, by the way, a nucleotide, right? So what is a nucleotide? A nucleotide is simply a nitrogen base that's attached to a sugar molecule that is attached to a phosphate, okay? And we call this whole structure here, we call this a nucleotide, right? Now, suppose that we get rid of the phosphate, right? So we get rid of the phosphate. Now, what's it called? Well, it's no longer a nucleotide. There's no phosphate on it. Now, it's going to be called a nucleoside. Okay? So basically, a nucleoside is simply a sugar that is attached to a nitrogen base. We call it a nucleoside. Okay? So suppose we want to cut it down further and we break something called the beta-N glycosidic bond over here, right? So these two bodies are no longer attached to one another. Now simply this is a nitrogen base and this is a sugar, and remember the phosphate, we cut it off also, and that's that. So the basic building block of the DNA is, well, I can't really say that it's a basic building block, because to make a DNA you'd need a phosphate, a sugar, and a nitrogen base. However, textbooks, they like saying that the nitrogenous bases are the basic building blocks, okay? And so you would imagine that the synthesis of DNA would go something like this. You have a nitrogen base, and it's, you know, it's floating around, it's roaming around, it's looking right and left. Ooh, it finds a sugar, and it grabs onto the sugar. And now we call these two a nucleoside, right? Now, these two are roaming with one another, and then they find a phosphate, and the phosphate goes and it binds to it. Now it's called a nucleotide. So suppose I am the central connector, I am the sugar, right? I first grab a nitrogen base. Now I'm called a nucleoside. Me and the nitrogen base are called nucleosides. And we roam around and we find a phosphate. Now I am called a nucleotide, okay? So how do I build myself? Well, basically, we've got millions and millions and probably billions of nucleotides, right? And they start to attach to one another. So again, this is my phosphate and this is the nitrogen base. I am the sugar and there are billions of me. And then we start to stack on top of one another. We reach the point of oligonucleotide. Once we become more than 20, 25, now we're polynucleotides. And once we form a whole DNA molecule, we get this, a DNA molecule, then it goes and finds a complementary DNA strand, another strand, and you get the double-stranded DNA, okay? Now, of course, this is not enough, so we're going to be talking about the importance of the sugar and some characteristics about it. And I'm not doing this because I like the pentose sugar or the ribose sugar and I'm in love with it and I'm going to tell everyone about it. No, I'm doing this because there's always clinical significance behind it. Then we're going to talk about the nitrogenous bases. We're going to tell you how to identify them, how you, as a medical student, could tell, for instance, a purine from a pyrimidine, or an adenine, guanine, from a thymine, from a cytosine, from a uracil, okay? Okay, so listen, before we dig into the sugar, the nitrogen base, and the phosphate, terminology is important, people. I like terminology. It makes my life much more easier, right? So DNA and RNA. What do they mean? And of course, you either know it or you do like a small Google search and you come back, you say it's deoxyribonucleic acid and this is ribonucleic acid. And of course, you'd be correct, but what does it mean, right? So DNA means deoxyribo. The N stands for nucleic, and the A would stand for acid. Okay, what about RNA? Well, RNA, the R stands for ribo, N for nucleic, and A stands for acid. So, looking at it, you would come to the conclusion that the only difference between a DNA molecule and an RNA molecule is the presence or absence of this word here, deoxy. And if you watch the lecture on the suffix and prefix that we're going to talk about, you already know that deoxy means we're removing something, right? So deoxy is Latin word that means lack or without, right? Without what? Without oxy. What's oxy? It stands for oxygen, O2. Where? On the ribo. What's a ribo? A ribo is a ribose sugar. And again, if you watch the lecture on prefix and the suffix, you'd know that O's is a sugar, right? So RNA, it has a ribose, remember O's, but it is not lacking an oxygen group. And basically, this is the main difference between DNA and RNA, the presence or absence of an oxygen on the sugar, okay? So, and by the way, it's very important when we reach sugar, when we start talking about the sugar, the ribose sugar that makes DNA or RNA, I'm going to tell you exactly why DNA must lack an oxygen and RNA must never lack an oxygen, okay? Don't worry about it. Now, we just want to analyze the word, the acronym, right, and understand what's going on. Okay, so great. So we know where the D came from, and we know where the R came from. They simply refer to the sugar, right? What about N? N refers to nucleic. Why nucleic? Well, in the 1800s, scientists came, they had a microscope, they looked under it, and they found DNA in the nucleus. And so they gave the word nucleic, right? What about the RNA? Listen, the RNA, 30 years later, 40 years later, something like that, they looked under the same cell, well, not the same cell, but you get the idea, right? And they found an RNA molecule inside the nucleus. And so they came to the conclusion that, yeah, this is nucleic, and the DNA is nucleic, right? However, later on, we found out that also the RNA can be present in the cytoplasm. But by the way, if you do not know what a cytoplasm is, this is the cytoplasm, right? This is the nucleus here, the nucleus, and this is just the cytoplasm, Okay, anyway, they found the RNA, it was in the cytoplasm, but you know, they were stuck. Okay, great. So that's in, and that's in. What about A, acid? Well, that's simple. That's because DNA and RNA are acids, right? Not quite. Listen, I'm not gonna dive deep down the rabbit hole on this one, because, you know, I already have like a mini lecture on this. But bear with me, would you? It's going to be fast. Acid versus basic. There are two ways you can think about this. If we're talking about, you know, blood fluid, basically anything physiology, then we know that the pH is going to be less than 7.4. It's going to be acidic, and if it's more than 7.4, it's going to be basic, right? So acidic, basic, that's great, but does not explain the DNA or the RNA why there's an acid here, okay? So there's also a biochemical terminology, a biochemical concept or terminology. Basically, the acid in deoxyribonucleic acid or ribonucleic acid refers to, or it's called acid because of the phosphate group, right? Why? Listen, it's something, it's like pKa, don't worry about it. Basically, the phosphate has the ability, you see it has a positive charge and a negative charge. The phosphate donates its positive charge, and therefore, under, of course, normal physiological pH conditions, it gives away a positive charge, a proton, right? Proton is a positive charge. And therefore, because it donates a positive charge, because it throws away a positive charge, we call it acidic, right? You have stuff like the amino group, right? To clarify this, the phosphate was happy, it was neutral, nothing going on, then it dumped its proton, and now it's called a phosphate PO4 negative, right? Why? Because it donated it, and so this chemical group in biochemistry is called acidic. NH2 is an amino group, same story. It had a positive and negative, it was neutral, it was happy until it found a positive proton, and then it accepted it. And so we call it basic. That's all. So DNA, the acid, RNA, the acid, they both come from the phosphate. Now, I don't know if you noticed this or not, but by simply understanding the name, we now conclude deoxyribo, so there's a sugar, and I really like to change it up a little bit, because I think that it should be called actually deoxyribo nitrogen base acid. Why? Because, you know, this stands for the sugar, the ribose sugar, which is deoxygenated, right? So it doesn't have an oxygen group. And the N for the nitrogen bases, and the acid simply refers to phosphate. That's it. RNA, same thing, right? So sugar, right? The ribose sugar, the nitrogen bases that make the RNA, and the acid, which is simply the phosphate. And that's all. Okay, so this is what the word really means. Okay, so let's start with the central connector, shall we? The central connector, which is the sugar, which is going to be the ribose sugar. Okay, now here we have the, you know what? Let's make them two different colors. Here's the DNA, and here's the RNA. Why does the RNA get to be blue? Yeah, it doesn't deserve to be. Here, and the RNA, right? Now, I'm going to draw both pentose sugars. I'm going to tell you why we call them pentose. I'm going to ask you, what difference do you see in them? At this point, you have a good eye. There's no oxygen group on the sugar or the ribose sugar that makes the DNA, while on the RNA, there's going to be a hydroxyl group. Remember, hydroxyl, OH. So it has an oxygen, the RNA is sugar. The RNA ribose has an oxygen, the DNA lacks it, okay? Now, before we talk about why, let us talk about the numbering system of the ribose sugar and why it's important, right? So the ribose sugar characteristics, number one, the numbering system. When we count the carbons, we're gonna say 1 prime, here's a carbon, 2 prime, 3 prime, 4 prime, and 5 prime. The same thing goes, of course, for the RNA, right? So why are they, first of all, why is it called prime? Why prime, right? Quite basically, the nitrogen base, remember, the sugar is a central connector. The nitrogenous bases are going to connect to the number 1 prime carbon, right? Now the nitrogen bases are rings themselves, just like this. And so we count them 1, 2, 3, 4, 5, 6, for instance. So to avoid confusion, we simply refer to the numbers on the sugar, on the ribose sugar, as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And great. So we spent a lot of time talking about numbering a goddamn sugar. Now what? Now all of them are important except for 4 prime. We don't care about it. How are they important? Now remember, we said that the sugar is a central connector. 1 prime is going to connect to the nitrogen base. 5 prime is going to connect to the phosphate group. 2 prime, well, basically 2 prime tells us the difference between DNA and RNA, right? So 2 prime, if there's an oxygen group, then it's RNA. If it lacks, if it's deoxygenated, then it's going to be a DNA. What about 3 prime? 3 prime, basically, this structure right here, remember we said this is going to be a nucleotide. If there's another nucleotide and it wants to attach this nucleotide to make an oligonucleotide and then a polynucleotide, then a DNA, they're going to attach themselves on the 3 prime, okay? And we're gonna talk about that, don't worry. So this is the numbering system. Number two, okay, so central connector, right? Central connector. Right, great, okay. Number three is why does the DNA lack an oxygen on the 2 prime end? Quite basically, listen, this is what happens. Here's an RNA, right? Here's an RNA. And the RNA, this is the backbone of the RNA. So basically the backbone is going to be the sugar and the phosphate, right? Now, because it has an oxygen here at the 2 prime end, here's the 1 prime, by the way, and 2 prime, 3 prime, 4 prime, and 5 prime. Now, because it lacks an oxygen group at the 2 prime end, what's going to happen is that it causes self-cleavage. It is very reactive. The mere presence of this oxygen right here is going to cause the RNA to go and attack itself and undergo something called hydrolysis. Now, hydrolysis, remember, so water breaking. This is an RNA, and I put a drop of water on it, it breaks, right? And so you'd imagine that the RNA is not going to survive for a very long time. Now, what about the DNA? Well, you see, the DNA has to survive for a very long time. As long as the cell is living, the DNA must be very chemically stable. How do we achieve that? Well, we simply remove the oxygen that was present here. And that's why the oxygen, or that's why DNA lacks oxygen. Okay? See, DNA is the genetic blueprint of the cell. It has to survive the entire life of the cell. And sometimes, if we're talking about germ cells like spermatocytes or oocytes, it has to even outlive them. It outlives the original human, right? And that's why it lacks the O2 to keep it chemically stable. What about RNA? We know that the cell has to be energy efficient, right? The cell is a very stingy subject, by the way. It does not like to use energy when it can, conserve it. So why would it make something? Why would it make a structure for a couple of minutes, half an hour, whatever, and then that structure goes and degrades itself. Well, think about it this way. Suppose this is a cell, right? And here is a nucleus, and of course, the cell is very happy. Why is it happy? It has its RNA, it has a messenger RNA, and this mRNA is making proteins, right? Now, suppose that this mRNA doesn't degrade itself. Suppose that it lacks an oxygen group like the DNA does. So the mRNA, the only thing it knows to do, or one of the most important functions of it, is to make proteins. And it's going to keep on making proteins and making proteins and making proteins, and eventually the cell dies. So RNA is chemically unstable. It has self-cleavage. It destroys itself. DNA, on the other hand, has to live for a very long time. Okay? So why did we spend so much time understanding the deoxyribose sugar or the ribose sugar? Oh yeah, another point before I go there. Listen, the ribose sugar is also called a pentose sugar by some textbooks. Penta, by the way, pentose or penta means 5. Penta means 5. Why is it 5? Because it has 5 carbons, right? It has 5 carbons, so we call it pentose. Of course, pentose simply refers to how many carbons it has. There's a lot of sugars that are also pentose. Xylose is considered pentose. Arabinose is considered pentose, as is ribose, right? And notice the ose suffix. Xylose, arabinose, ribose, right? So it's a suffix, means they're sugars. That's all. Okay, so again, why did we spend so much time talking about the ribose sugar? Imagine this. There are some tumors, or let's take an example about the herpes simplex virus, right? So herpes simplex virus, it's a DNA virus, right? And it's the way, by the way, most species, or all of them in fact, except if it's an RNA virus, make their DNA is extremely close to how we make our DNA, right? So, and this herpes simplex virus, it infected this person. And now we go and we give something like Cytarabine. Cytarabine is a drug. The difference, it resembles ribose sugar, but it actually contains arabinose sugar. The arabinose sugar doesn't look quite like this, you know, it has some, maybe the structure is different, maybe it lacks an oxygen group here. And this is just as an example. We're going to talk about this when we reach pharmacology, right? But just bear for me for a minute, just bear with me. So suppose that we give a drug and we remove the hydroxyl group from here, we remove the oxygen and we left this. And so the virus would come and it's trying to build its own DNA and takes it up, and by the time it takes it up and it incorporates it into its own DNA, and now it's going to take another nucleotide, right? And it wants to stick it on here. But wait, where's the oxygen? This is called chain termination. This is how, one of the ways where we kill viruses, and by the way, one of the ways where we can use drugs in treating certain leukemias. Stuff like Cytarabine, right? Cytarabine is a drug that's used in acute myeloid leukemia by interfering with the DNA synthesis and replication. Okay, basically, again, Cytarabine, vidarabine, fludarabine, right? Now, listen, look at the suffix, arabine, right? So basically, the sugar is not a ribose sugar in these drugs. They just resemble the ribose sugar, in which case, if it's a cell that has cancer, for instance, like an acute myeloid leukemia or chronic lymphocytic leukemia, they're going to take up that sugar into the DNA, because remember, cancer cells, all they want to do is divide, right? Now, if you cause chain termination, if you prevent the DNA from, elongating itself, then the cell never becomes, and you try to control the cancer. Okay? I think that's all we have to talk about for the sugar. Okay? Yeah.
Okay, now it's time for the nitrogenous bases. The NB, right? Again, nitrogenous bases. Why do we call them nitrogenous bases? Well, nitrogenous because they contain nitrogen at different carbon numbers, and bases because, remember, they accept a proton and we call them bases. Okay, so what do we need to know about them? Nitrogenous bases. They're subdivided into purines and pyrimidines. There's a pyramid. Okay, I'll write it down. I don't want to be lazy. Here's a pyrimidine. Right? Here's the pyramid. Here's a pyramid. Okay, anyway. So, purines and pyrimidines, what's the difference? A purine has 2 rings. A pyrimidine only has 1. So, how are you going to remember this? Easy. Pu, here's 1 ring. Rene, and here's another ring. So they have 2 rings, the purines. What are they? They're adenine, or it could be guanine. What about the pyrimidines? The pyrimidines have only 1 ring, and the way I remember this is, if that has 2 rings, this has 1. Okay, it's a pyramid, 1 ring. So anyway, cytosine, it could be uracil, and it could be thymine. By the way, don't say thymine, because that's a vitamin, okay? Thymine. They're very close to one another, but anyway, you get the idea. Now, how do students, a lot of them remember this? Purines, they say it's pure as gold, and the pyrimidines, they simply say cut. Okay? Now, unfortunately, at this point, me and you are parting. It's time for the tablet. It's going to make things faster. It's going to make explaining this much more easier. Okay? So to the tablet. Easier. Okay, so a quick recap on the nitrogenous bases. In DNA or in RNA, there are going to be purines, right? And there are going to be pyrimidines. How do you tell the difference? First of all, before we go there, the difference between DNA and RNA, there's only 1. In DNA, it's going to be thymine. In the RNA, it's going to be replaced by uracil. Okay, so DNA contains thymine. RNA, instead of thymine, it has uracil. Okay, and that's the only difference between the nitrogenous bases. Now, the nitrogenous bases themselves, they could be either purines, which means there are 2 rings, right? Or they could be pyrimidines, meaning that there's only going to be 1 ring. Okay, and here's a table to quickly recap it. So adenine is going to be a purine. Guanine is also going to be a purine. Cytosine, thymine, uracil, and all of these 3 are going to be pyrimidines. Notice that thymine is only present in DNA, while uracil is only present in RNA. And here is a visual so you can understand it really. So purines, again, they have 2 rings, 1 ring, 2 ring, and pyrimidines, they only have 1 ring. Now, one of the things you're going to encounter in the exam is, well, again, the purines. By the way, there is a, students use it, they say that purines are pure as gold, to remember the purines, which are adenine and guanine, right? So pure as gold, adenine, guanine. Pyrimidines, again, only 1 ring. You've got cytosine, thymine, and uracil. And the mnemonic is cut, right? So cytosine, uracil, and thymine. Now, why is this important for us to know? Now, one of the ways the USMLE asks about it is that they could present you with a nitrogen base. And then they expect you to know which nitrogen base it is, okay? So again, if it's 2 rings, it's going to be a purine. If it's 1 ring, it's going to be a pyrimidine. And that's nice, right? So how do you tell? It's very easy to tell a purine from a pyrimidine, but how do you tell 2 purines from one another? For instance, over here, we have the adenine and we have the guanine. And they're going to give you the whole chemical structure of it if they ask about it.
First of all, I want you to forget about all of these. They are not important, okay? Not important. What is important is the carbon number 6 position, which is, in the case of adenine, this one right here. So in purines, if you see an amino group, this is an amino group, by the way. There's an amino group. If you see it at the 6 carbon or the 12 o'clock position, some examinees call it. So here's carbon number 1, 2, 3, 4, 5. This would be carbon number 6. And you look atop it, and if you see an amino group, you know this is going to be adenine. What about guanine? So great, here's ring number 1, ring number 2. Where's carbon number 6 or the 12 o'clock position? And here it is. What does it have? Does it have an amino group? No. And therefore, this is going to be a guanine. Okay. What about the pyrimidines? So what about the pyrimidines? Pyrimidines, again, they have one ring. So here's one ring, one ring, one ring. How do we tell them apart? How do you tell cytosine from thymine from uracil? First of all, again, we look at the 12 o'clock position or carbon number 6, right? If it has one ring and has an amino group on it, here it is, here's the amino group, then this is going to be cytosine. We do not care about all these other structures. Now, what if it's, okay, so basically uracil and thymine, they do not have an amino group. So how do you tell thymine and uracil apart? Simple. Thymine has a methyl group, and usually it's written like this, HC3, right? So if you see a pyrimidine, meaning there is one ring, and you see an amino group, then this is cytosine. If you see a pyrimidine and you see, if you see one ring, it means a pyrimidine. If you do not see an amino group, then that means that could be either uracil or thymine. How do you differentiate between these two? Well, again, you look at the carbon number 5 here, you're going to see a methyl group, and that's all. So the only difference between uracil and thymine really is the presence or absence of the methyl group. So now we're faced with two questions. Number 1, why is there no thymine in RNA? And number 2, why is there no uracil in DNA? Let us start with the simpler explanation, which is why is there no thymine in RNA, right? Now, chemically, uracil is like thymine, but without the methyl group, okay? And the reason that RNA really does not contain thymine, first of all, thymine making uracil is simpler. It's cheaper. Remember that we said that the RNA, because it has it has an oxygen at the 2' carbon of its sugar, of the ribose sugar, this means that's going to be degraded quickly, right? And therefore, the cell, being energy efficient, doesn't really want to use extra energy putting methyl groups on every base, okay? And the second reason is for speed. Because RNA is going to be produced, it's degraded constantly, so of course simplicity is going to be always favored. Okay, now that answers the first question. Okay, it's very easy. That's why we don't have thymine in RNA. Basically, energy, efficiency, and speed. So let's go to the second question, which is why is there no uracil in DNA? So there are 3 reasons for this. Number 1 is something called cytosine deamination problem. Let's leave it for now. Number 2 is stability. Why? Because thymine, remember, we said thymine has a methyl group, right? And what this methyl group does is it makes DNA more stable, it makes it more resistant to mutation, okay? Again, we said that DNA, it's the genetic blueprint of the cell, and therefore we have to protect it at all cost. And which explains really point number 3, which is longevity versus disposability. DNA is a permanent storage, and therefore it needs extra protection. RNA, remember, it's a temporary copy, it's a temporary message, and therefore we use cheaper material to make it, okay? Like uracil, which in fact is more stable. Now, the important point here, the one I want to talk about, is the cytosine deamination problem. Now, remember people, we said that to always look at the title, it explains a lot. Deamination. Remember when we said deoxy and we said that the DE simply is a prefix that means lacking or without, right? And amination, or it refers to the amino group, which simply means NH3+. Okay, so deamination means we take the cytosine and remove the amino group, and now we are left with something called uracil. And that is quite problematic. So why is this problematic? Listen, let's draw a strip of DNA, right? And it has nitrogen bases. So it has adenine, guanine, cytosine, thymine, adenine again, for instance, cytosine, thymine, okay? Now, sometimes a mutation occurs that is called spontaneous cytosine deamination, right? And we talked about this. We said the cytosine, it spontaneously loses the amino group and it becomes like this, right? And suppose that the C here underwent that, and now you have a uracil. And so we have these little enzymes, clipper enzymes, that they come in and they see that, you know, there's a uracil here, and uracil shouldn't be present in DNA. They cut out this portion, they remove the uracil, and then they reinsert the cytosine again, and everything's fine. Okay? Now, suppose that uracil was in fact present in DNA. So let's draw another strip of DNA, and let's put nitrogen bases, adenine, for instance, guanine, thymine, cytosine, uracil, guanine again, cytosine, uracil, for instance, okay? And so let's suppose that this C over here underwent spontaneous cytosine deamination, and now it's transformed into a U. And so this repair enzyme, it comes in, right? And, you know, it's checking, okay, you're good. It sees a uracil, fine, you're where you're supposed to be, guanine, and then it comes here and sees a uracil, okay? And since, well, it thinks to itself, well, uracil is normal, it's present, and so it just keeps on moving on. And it does not fix this uracil right here. So what happens is, now this uracil here is going to be a hotspot for mutation. It can cause severe problems. Okay, and this is what is called cytosine or spontaneous cytosine deamination, and that's why one of the more important reasons why we do not use uracil in DNA. So, can uracil be present in the DNA? In fact, it can. Two problems, or two ways it can occur. First of all, is the spontaneous cytosine deamination. Again, we said cytosine loses its amino group and then becomes uracil, and we talked about this. Number two is the replication errors, right? Basically, one of the enzymes that's involved in DNA replication is called the DNA polymerases. It's a very important enzyme, and sometimes it makes a mistake and it incorporates deoxyuridine monophosphate instead of deoxythymidine monophosphate. Okay, so this is why, yeah, uracil can sometimes be present in DNA, and it is also very problematic. It causes mutations if it's not fixed. However, it usually is, because remember, cells always have specialized repair systems. Okay, in this case, it's something called a UDG scan, or a uracil DNA glycosylase. Not important for us here. We're going to talk about this when we go over mutations. All right, but basically, it removes the uracil base, and then you know, base excision repair enzymes come in and they fix the problem.
Okay, so far we went over the ribose sugar and the nitrogenous bases. Now, where do we go from here? Well, we want to connect them. Remember, we're making a DNA, so we want to build a nucleoside. To build a nucleotide, then to build an oligonucleotide, then a polynucleotide, then a single-stranded DNA, and eventually a double-stranded DNA. So, back to the tablet.
Okay, so now we have our sugar, our ribose sugar, and we've got our purine and our, of course, pyrimidine. right? And now we want to connect these two with one another to form something called a nucleoside. Now, the way I'm going to do this is that I am going to draw for you a certain structure. And before I say it, I want you to pause and think about the structure I have just written, okay? So, basically, first of all, what happens is, on this sugar right here, remember 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Where does the nitrogenous base, whether it be a purine or a pyrimidine, where does it link? It links at the prime number 1, and the linkage, if you want to know it, is called beta N glycosidic bond. Okay? Now, to the juicy stuff. What's this? Well, it is the 12 o'clock position, or the carbon number 6. Remember? 1, 2, 3, 4, 5, 6. Is there an amino group? No. How many rings? It has 1 ring, it has 2 rings. So what is it? And don't say guanine. I mean, the nitrogen base is guanine. However, remember, it's going to be connected at the 1 prime to the ribose sugar. And so, what's it called? Well, you can't really tell yet. Why? Because it could be either guanosine or it could be deoxyguanosine. So, how do we know? Well, if there's a hydroxyl group at 2 prime, then you know that this is an RNA, and so it's called guanosine. If there is no oxygen here, only a hydrogen, at the 2 prime on the ribose, it's called deoxyguanosine. Okay, what's this? In fact, you know what? I'm not going to draw anything. What is this here? This is 2 prime. Here, again, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, just so you don't forget. And this is the carbon number 6 on the nitrogen base. What is it? And listen, I know there's going to be chemical structures here. However, remember, there's nothing on 6, there's nothing on 5, or if simply does something like this, this is a uracil. Okay, the nitrogen base is called uracil. The whole structure here, since it's an RNA, is going to be called uridine. Okay, let's draw another one. Okay, here. Here's an amino group. What's this structure? Well, since at the 12 o'clock position, or carbon number 6, first of all, 2 purines, 1 purine, 2 purines. There's no oxygen here, so it comes from DNA, so it's deoxy. Deoxy what? What's the structure? And it's adenine, so it becomes deoxyadenosine, right? Okay, what about this? What's this structure? Here. I'll tell you what the structure is. This structure is wrong. Now, you may be tempted to say something like deoxythymidine or thymidine, but remember, this comes from RNA, and thymine is never present. And so, this is a wrong structure. Okay? It's never present. The thymine is never present in RNA. This is an RNA, right? Now, if I did something like this, if I remove the methyl group here, and I give something like this, now this is going to be uridine, right? Okay, what's here? One ring, 2 rings, and there's an amino group. So this is adenine, right? Now since it's connected and the number 2 prime has its oxygen, has a hydroxyl group, and therefore this is going to be called adenosine. Okay, what about this? What about this? Or, you know, what's this? This is deoxyribose, right? Now, I'm going to draw them again. I'm sorry, I know, but it's very important that you understand this. Now, let's start with the DNA, then we're going to turn it into RNA, right? Now, what's this? This is deoxy, remember, because there's no oxygen on the 2', so it's going to be called deoxyadenosine, right? Okay, what about this? What's this? So, on the 2', there's no oxygen, so it's deoxy, 1 ring, 2 ring, no NH2 group, no amino group, so this is going to be deoxyguanosine. Great. What about this? What's this going to be? It's going to be, yeah, deoxycitidine. What about this? You're correct. That's going to be deoxythymidine, or sometimes we simply refer to it as thymidine, right? Okay. What about this? And I hope you didn't say deoxyuridine, because remember, uracil, this is uracil, because remember, there's no NH2 group, and there's no methyl group, so it's going to be uracil. However, technically, it could be found in DNA, but it would be an error, and it would be wrong, okay? So the correct manner to write this would be to add an oxygen group on the 2', right? So it becomes uridine. Great. So, what if I do this? What's this now? Remember, now it's part of RNA because there's an oxygen here. And it's going to be called, adenosine, right? So, adenosine. What about this? Now it's going to be called guanosine, right? Now, notice in the purines, there's the sine suffix. What about this? 1 ring, amino group. This is going to be called Cytidine, right? What about this? Careful now. This is thymine. It cannot be present in RNA, and so the structure is wrong, right? So, and this, of course, is going to be uridine.
Now we come to the juicy part, the clinical significance of nucleoside. And so great, now we absolutely understand where the nucleoside came from, right? And we talked about them. We said we have adenosine, guanosine, cytidine, thymidine, uracil. Why do we care? Why do we care about the nucleosides? In fact, they're extremely important, all of them. Let's start with the most important one, which is adenosine, right? So, clinical significance of adenosine or deoxyadenosine, same thing. So, point number 1. Adenosine in adenosine deaminase deficiency. Now remember, we said adenosine was simply a ribose sugar, right? That is connected to a nitrogenous base, okay? And this nitrogenous base has an amino group. Remember, 2 rings, 1 ring and 2 ring. Sorry for the way I draw them, but anyway, okay? There's an enzyme. This enzyme is called adenosine deaminase. What it does is, look at the word. Remember, adenosine, so basically it's working with something, which is the adenosine, deaminase, so it's removing the amino group, aminase, right? So it removes this group. Now, if we remove this group here, okay, probably shouldn't have done that, but okay, here, there you go. Right? So if it removes this, now this structure over here is no longer called adenosine. Instead, it's called adenosine. Right? Adenosine basically it goes through this pathway. It's called a purine salvage or a purine degradation pathway. The inosine becomes something called hypoxanthine. Here, hypoxanthine, and then the hypoxanthine becomes something called xanthine, right? And the xanthine eventually it becomes uric acid. And uric acid is secreted out of the body by your urine. Now, if you're making too much uric acid or you're under secreting uric acid, you get a condition called gout. Right? And that's where, you know, basically a lot of the patients, it's very painful, right? They say, oh, my toe, it's killing me, it's hurting, it's inflamed, whatever. The purine degradation pathway is not the reason we're talking about this, however. There is a condition, and this condition is called adenosine deaminase deficiency. Basically, this enzyme, imagine a patient has very, very low levels of it. And so, okay, and since there's low levels of this enzyme, that means that adenosine, the amino group on the adenosine, is never going to get removed, right? So what? Let it be, right? That's what you might think. And where's the big deal? So let's have the adenosine just, you know, floating around and everything's going to be great. Well, there is a problem. Once adenosine starts to accumulate, it's going to give us a very high amount of adenosine and something called DTAP. Oh, sorry, actually, not DATP, sorry. It's like a mental block at this stage. DATP, deoxyadenosine triphosphate. So basically you have a very high level of adenosine and deoxyadenosine triphosphate. Don't worry about this name, I'll explain it in a moment, okay? But anyway, now once they start to accumulate, they become very toxic to the cells and especially lymphocytes. Lymphocytes are one of the groups of the white blood cells. They help us fight off infections, correct? So if they're toxic to lymphocytes, they're going to start killing the lymphocytes. Lymphocytes are going to start to die. And that means that the patient is going to become susceptible to having recurrent infections, okay? And this leads to a disease called SCID. SCID simply refers to severe combined immunodeficiency. Well, one of the reasons would be this. It can be due to other reasons, by the way. SCID is like responsible for about 10 to 15%, the adenosine deaminase deficiency is only responsible for 10 to 15% of the cases of SCID. A lot of them are actually X-linked recessive, but we'll talk about it when we reach immunology. By the way, academia loves severe combined immunodeficiency because it was the first disease to actually be cured by gene therapy, right? Now, so this is the first clinical significance of severe combined immunodeficiency. Number two. So its role in cardiology, right? Red for the heart. Adenosine has two major roles. It's actually used as a pharmacological agent in some cardiac conditions. Number one is something called SVT, supraventricular tachycardia. What happens in supraventricular tachycardia is that the heart rate is going to be blowing through the roof, right? The patient comes in, usually the patients have had this episode before, okay? So they're kind of expecting what's gonna happen. One of the things that the patients with SVT are always complaining about is something called an impending death. They feel that they're going to die because that's what SVT that's what it feels like. It feels like death. Okay? So he comes to the emergency room, or she comes to the emergency room, and she's like, "Doc, please, I'm gonna die, etc. I'm just gonna throw myself on the bed over there. You get your adenosine and give it to me, and we'll forget that this ever happened." Right? By the way, Doctors are the most thanked and the most sued people on the earth. So, Anyway, SVT, you give the patient adenosine. Why do you give him adenosine? Because once you inject adenosine into the patient's arm, it's going to cause a decrease in something called the AV conduction. Again, listen, I know you probably don't understand a lot of this. Maybe you're a first-year medical student. Maybe you're pre-med, whatever the case is. All I want you to know is that adenosine, we use it a lot. It has a lot of clinical significance. So we can use it in supraventricular tachycardia. Where else can we use it? We use it in a pharmacological stress test, right? What's a stress test? A stress test is in the cardiologist's office. They usually have a treadmill. They put the ECG on the patient, and then they ask the patient to walk to see if there's any areas of ischemia, meaning that there's a decrease in blood flow to that area. And so he gets up and he starts to walk, and the cardiologist would look at the ECG and determine if there's ischemia or if there's unstable angina or whatever, right? Now, what if the patient comes in, he's crippled, or he's an older patient that has maybe osteoarthritis, right? Any reason that makes maybe has a broken leg, right? So how am I going to do a stress test? Well, I give him adenosine. Why do I give him adenosine? Because adenosine is a vasodilator. That means that the vessels, especially those around the heart, when I give adenosine, they're going to dilate, they're going to expand. So, it becomes like this. It expands it, right? Once we talk about pharmacology, I'll explain it in detail. For our purposes right now, just know adenosine is used as a drug in stress test. Okay? Point number 4. Okay, so first of all, apnea of the newborn, right? Or what happens is that some premature babies, right? In premature babies, the respiratory centers and brain stem, they're not really well developed, right? And adenosine is kind of, it likes to be calm, it likes to relax the body, etc. And so one of the things it does is, it causes a depression in respiration. So what do? We give the babies caffeine. Please never give a baby a coffee. You're going to kill him. We give it in a form called caffeine citrulline, I think, right? Basically, it's in a very narrow therapeutic dose. What happens is, listen. Now, if you've ever played Elden Rings, there is something called the mimic, right? Basically, what a mimic is something that looks exactly like you. It functions like you. It talks like you. It walks like you, right? A mimic. Caffeine is a molecular mimic at a molecular level, at a very small level. It's a mimic to adenosine. Adenosine binds to receptors in the brainstem, the respiratory center, and causes depression of respiration. That's why when we are asleep, the breathing quality, it kind of decreases, it becomes more shallow, it becomes slower. And in babies, in premature babies, where they have apnea means they're like, they can't really, the diaphragmatic contractility, the respiratory centers are depressed, basically. So we give them caffeine. Caffeine, since it looks like adenosine, is going to have a fight with adenosine. Suppose that this is a receptor for adenosine, something that engulfs adenosine to make adenosine give its action. What happens is, Mr. Caffeine is going to come in and push out adenosine, get lost adenosine, and it's going to sit eventually in his place and causes stimulation of the respiratory centers. And so it increases the diaphragmatic contractility, it increases the respiratory rate, and the babies have less apneic episodes and they get better oxygenation. Most of you, probably all of you, drink coffee. right? So why does coffee keep you awake? Again, what happens is, you see, again, we said that adenosine itself is one of those molecules that binds in specific areas in the brain, and it causes a sense of calmness, drowsiness. It makes you want to sleep. Now, caffeine looks like adenosine. And when you drink enough caffeine, it's going to compete with adenosine for the receptors. And so it pushes away the adenosine and sits in the receptors, and now you're awake and you're alert and you're watching Draminor Medicals and you're happy. The thing about adenosine is adenosine never goes away. While caffeine is sitting there in its receptors, adenosine is just accumulating in the background, right? It's watching, it's waiting, because eventually adenosine knows that caffeine is going to go bye-bye. It has the half-life of caffeine is maybe like, 8 hours, 5 hours, 6 hours, something like that. So after 5 to 6 hours, and you're alert and you're happy, suddenly caffeine goes bye-bye, and adenosine, the huge amounts of adenosine that have been accumulating in the background, rush into the receptors, and now you get what is called a caffeine crash. And that's why you get a caffeine crash, that's why caffeine keeps you awake, and that's all. Okay? And that was the clinical significance of adenosine. Okay, let's look at guanosine. Okay, so guanosine analogs, right? We're going to use the green color because, it kind of fits the theme. Guanosine, right? Now, this is biochemistry, great. We talked about, in adenosine, we talked about the immunology, we talked about cardiology, we talked about pediatrics, we talked about sleep medicine, right? Now, we're talking about pharmacology and virology. We're going to see how exactly we use guanosine analogs to kill viruses. Number one, so guanosine analogs, again, there are going to be acyclovir, it's going to be ganciclovir and ribavirin. Let's start with acyclovir. Acyclovir. Now, to understand how acyclovir actually works, let's look at the normal structure of guanosine, right? Or deoxyguanosine. Because acyclovir, usually we give it to patients that have herpes simplex virus, right? So, this is the ribose sugar. Again, this is going to be a deoxyguanosine, right? Here's the 3', here's the 2', and attached to it is going to be a guanine nitrogen base, right? So 1 ring, 2 ring, and here's an oxygen. There's no amino group because that would make it adenosine, right? Now, this is the normal structure of guanosine. Acyclovir, you know what, I'm not even going to draw it anymore. Listen, acyclovir, you see this 3' here? This is what acyclovir is. It's the exact same structure, but without the hydroxyl group. Now, if you recall, if you remember, we said that when the nucleotides, remember I said I was a nucleotide, and once, or I am a sugar, and I grab a nitrogen base, and I grab a phosphate, and now I'm a nucleotide. And the way I build myself is, let's say that this is the 5' end, my feet are the 3' end, another one of like nucleotide, whether it be adenine or guanine or whatever, they come and they stick themselves underneath my feet because it's the 3' end. Once acyclovir enters the cell, here's a big fat cell for you, okay? And here is acyclovir, and so acyclovir enters the cell. Now, inside the cell, what's going to happen is that the virus that's inside the cell, here he is, he's looking all mad, and he's, really bad virus. So he's going to take the guanosine, and he's going to put a phosphate on it, right? Or the acyclovir, sorry. Of course, the acyclovir is nothing but a guanosine with a missing hydroxyl group on the 3'. And so he's building his DNA. So far, let's say he built he put an adenine, then he put, a thymidine, and now he went and he put, or he thinks it's a guanosine, but in fact, it's going to be a cyclovir. So it's like a fake guanosine, right? And he's happy. He attached this to the 3', he attached this to the 3'. Now he goes and he takes, let's say he takes, what does he take? Let's say he takes thymine again, and he wants to attach it here. But behold, it has no 3', or it has no hydroxyl group on the 3'. And so what do we get? We get chain termination, DNA synthesis halts, it stops, and you can imagine this happening on thousands and thousands and thousands of cells that contain this bad boy. Eventually, he won't be able to replicate himself, and he dies. The patient gets better. That's a cyclovir. Okay? Now, what's the other drug? Gancyclovir, right? Now, I'm not gonna redraw anything here. I'm just gonna move a cyclovir and this, and I'm gonna put gancyclovir, right? Why? Because gancyclovir works on a virus called cytomegalovirus. Basically, cytomegalovirus is more resistant to a cyclovir, and so, they developed a new drug that is called gancyclovir, which has more potent activity against the cytomegalovirus. Now, gancyclovir, it in fact does have a hydroxyl group. The only difference is the hydroxyl group is shaped kind of funky. It's weird the way it's shaped. So it doesn't really cause immediate halting of the DNA synthesis. However, it kind of slows it down. Eventually, if enough of the gancyclovir come in, they are going to cause halting in DNA synthesis. Okay? A very important point, by the way. Listen. About acyclovir and gancyclovir. About both of them, in fact. Listen. The main difference, and gancyclovir causes severe side effects. One of them is neutropenia, which means it causes the white blood cell levels in the body to go down. Acyclovir doesn't do that. Why? Acyclovir, the only thing that can activate it when it's inside the cell is the viral kinase. Okay? And therefore, if a cell is not infected with the virus, even if the acyclovir comes and enters it, it's never going to be activated. There's no phosphate that's going to be put on it. And so it's not going to work. Gancyclovir, on the other hand, it can be activated by the virus, by a cell that's infected by the virus, or even if it enters a normal cell, it can be activated in there. And therefore, you would imagine that it can in fact affect cells that are infected with the virus and cells that are totally normal and totally healthy. Okay? Okay, so cytosine's up next. Let's get rid of the guanosine. Let's leave the cell, maybe we'll need it, right? So, the clinical significance of cytosine. The first drug here is something called Cytarabine. So what is exactly Cytarabine, right? Now let's make the cytosine, right? Again, 1', 2', 3', a hydroxyl group. Everything's nice, right? And of course, the nitrogen base, cytidine, here, with the amino group. And forget about that. Cytarabine is used in leukemias and in lymphomas, but mostly in acute myeloid leukemia. Why? Well, cytarabine targets the tissues that are rapidly dividing. And in leukemias, in any cancer, cancers are rapidly dividing as well. So how does it exactly work? How does this nucleoside work? Basically, listen, cytarabine, it looks like acetidine, However, the sugar that is used is an arabinose sugar. It's a pentose sugar, remember 5 carbons, but the structure of it is kind of different, right? For instance, first of all, it has a hydroxyl group on 2'. Okay, that's no problem. I mean, sure, it makes the DNA less stable, but what it actually does is, consider this is the cytarabine, right? And so it has the 2' hydroxyl group, 3' hydroxyl group. And then, fuck, I'm tired, I am. I'm really tired, sorry. Okay, anyway. So, the way cytarabine is done is that the hydroxyl group on the 2' end is going to be oriented in a manner that is opposite to the 3' end. So basically, now you have a hydroxyl group here and a hydroxyl group here on the 3' end, but look at the hydroxyl group, how it's oriented on the 2' end, right? And that means when the tumor cell, when the cancer cell incorporates it into its DNA, it puts cytarabine, and now it wants to go and bring another nucleotide, which is this one. Look, it's oriented, it's normal, right? And it wants to put it and stack them on top of one another. Look what happens. The mere presence, the orientation of the hydroxyl group on the 2' end of the cytarabine will not allow it, the geometry, let's say, the 3D shape of it, is not going to allow the incoming nucleotide to bind to it. So what happens? Chain termination. The problem with cytarabine and drugs like it is that since they target rapidly dividing cells, that means that they're going to target also cells that, are present in the bone marrow. And therefore, they're going to also affect the number of white blood cells. They're going to start to die. Remember, there is DNA synthesis termination. Platelet levels are going to decrease. Patients get something called mucositis. Basically, it's a gut inflammation. Why? Because the cells in the small intestines and the large intestine, they're always, turning over. They're always changing. They're rapidly dividing. Therefore, these patients, they get diarrhea, they get vomiting, etc. Low white blood cells, low platelet levels. Okay, now that was cytarabine. Number 2. Okay, so azacitidine and decitabine. Listen, very easy. Again, basically, you know what? First of all, you've got to understand something. There are some genes, like suppose this is a fragment of a DNA, right? And it contains genes. And these genes are going to be maybe rich in cytidine monophosphate, right? So CC, for instance, GAC, whatever. These genes, one of the important functions they have is that they are they're called tumor suppressing genes, right? So what does tumor suppressing mean genes? What does tumor suppressing genes mean, right? Basically, a tumor suppressing gene is a gene that makes proteins eventually that go and fight cancer cells. They do not allow the cancer cells to live and replicate, right? Now, why am I bringing this up here? There's an enzyme. This enzyme is called the DNA methyl transferase, or for short DNMT, right? So look at the name, DNA methyl transferase. What does it do, this enzyme? Basically, it takes methyl group, right? Remember, we said the methyl group is CH3, and it transfers it. Where does it transfer the methyl group? It takes the methyl group and puts it on the 5 carbon end, or the 5' of the cytidine, right? And you can imagine when this happens here, this process of methylating the cytidine or the cytosine causes it to become silent. Meaning that if there is a tumor cell and it's roaming around and it's like, yeah, I'm gonna kill this man, this woman, this boy, this girl, whatever. I'm gonna kill this body. And so since the tumor suppressing gene, this gene here, is silence, all it can do is just watch it while it goes and kills the body. Now, this is where azacitidine and decitabine come in. What they do here is these are cytosine analogs. What does that mean? It means, listen, it means they look exactly like cytosine, right? The only difference is at the 5' carbon here, at the 5' end. Instead of a carbon being here, they put a nitrogen base. So the drugs, what they do is we manufacture it in a way where over here at the 5' end, there's no carbon, instead there's a nitrogen. Okay, so what? So what? I'll tell you so what. So when the DNA methyltransferase comes and tries to put a methyl group right here, it cannot. It's like, well, where's the carbon? And Azacitidine and Decitabine, they're like, well, no carbon for you here, boy. It's only nitrogen. And so now DNA methylation, it's just looking there. It has its methyl group, and it's grabbing it. So, what do I do with you? We can't. Anyway, the end result is what? The end result is, since there is no methylation, that means that there is going to be no suppression of the tumor suppressing gene. This is not going to be suppressed, and therefore it's going to be active. And now, the gene itself, it pops up its muscles and starts fighting tumor cells. That's the idea behind Azacitidine and Decitabine. Very simple, right? Beautiful, beautiful. And by the way, don't worry about the tumor suppressing genes and all of that. We're going to discuss it in detail. We have a lecture about it. Just trust the process, okay? Okay, what else do we have? So, we're done with the ribose sugar. We're done with the nitrogen bases. We have one element left, and that's the phosphate. Okay? Now, phosphate. Is it important? It's probably the most important one so far. Listen, we need to know 4 things about phosphate. Number 1, phosphate forms what is known as the backbone of DNA. Or, well, it's and the sugar form the backbone of the DNA, but without phosphate, you don't get a backbone. I'll explain it, don't worry about it. Number 2, phosphate gives a negative charge to DNA. And this negative charge is going to be extremely important when we talk about DNA organization and how really the DNA itself interacts with something called a histone or histonoctamer, okay? It's very important. Number 3, we need phosphate for energy, or we use it for energy. Okay? Number 4, and I'll explain all these points, don't worry about it. Right? Number 4. I kind of forgot what number 4 was. Anyway, there is number 4. Just put that in mind. It'll come up. Now, you really want to know what number 4 is? Well, wait a minute. Gotta look at my slides. Oh yeah. Direction. Also extremely important. I'm having a lot of mental blocks today. Direction. What does direction mean? You're going to see this a lot. It's going to be 5' to 3'. Listen, I just want to say and tell you something. Maybe you think we're going into too much detail, right? But believe me, once we reach and we're talking about DNA replication, we're talking about translation, transcription, we are talking about mutation, it's going to be extremely easy for you because you can imagine every step of the process. Eventually, you're going to be so good at biochemistry that any exam you take, any professor that asks you anything is going to think to himself, this guy, he's a fucking legend, okay? And that's why we're taking so much time. I'm sorry, I know maybe you don't like it, but it's extremely important. Okay, so what do we start with? Let's start with the backbone, okay? Let's talk about the backbone and how phosphate comes into play into the backbone, right? So, here we go. Now, remember, we said that there's a nitrogen base, And this is a ribose sugar. And since we're talking about DNA here, not RNA, but put in mind, they're going to be made out of the same elements. They're basically made the same way. So at the 2' end, there's going to be a lack of oxygen. At the 3' end, there's going to be a hydroxyl group, right? Now, this and this, the sugar or the nitrogen base that is attached to the sugar is going to be called a nucleoside, right? Now, once we put a phosphate group on it, we call it a nucleotide. And this bad boy right here, now, depends on the nitrogen base, right? So it could be, for instance, okay, let me tell you about the nomenclature really quickly. The 3 things you need to know here. Mono means 1. Di means 2. And tri, it means 3. So if I have 1 phosphate here, I'm going to call it, and this is, well, you could call it a nucleoside by itself and add the phosphate. So basically, you could say that this structure right here, the ribose sugar plus the nitrogen base, which is, you know, a nucleoside, right? And since it has 1 phosphate group on it, we call it nucleoside mono 1, mono what? Monophosphate. Okay? If it has 2 phosphate groups, what do you call it? Well, it's no longer monophosphate, it's going to be called nucleoside diphosphate, right? If it has 3 phosphates on it, attached to it, now it's called a nucleoside triphosphate, right? So, let's say that this nitrogen base here is adenine, for instance. In this case, this nucleoside, since we put a name to the face, and it has 3 phosphate. Okay, you know what? Let's make it 1 phosphate. Here, nomenclature, right? Let's remove this. So, suppose this is going to be the nitrogen base. It has 2 rings. It has an amino group on the 12 o'clock position, right? So it's adenine. So what do we call this? We call this adenine mono, because 1 phosphate, adenine monophosphate, AMP. And you're going to see this. I promise you, you are going to see it a lot. What if it has 2 phosphates on it? Then it's adenosine diphosphate. If it has 3 phosphate, then we call it adenosine triphosphate, the ATP. The energy currency of the cell, right? We always say it's ATP. This is what ATP is. Remember when we talked about the severe combined immunodeficiency, the SCID, right, in immunology, and you said one of the reasons was adenosine deaminase deficiency, right? And you said that there's going to be of course, since this is a DNA, by the way, these all refer to RNA. Why? Well, adenosine monophosphate. If it was DNA, we'd call it deoxyaminomonophosphate, right? Or adenosine diphosphate, or deoxyadenosine triphosphate, right? Now, of course, this would be if it was a DNA, okay? If it's an RNA, then it's going to be AMP, ADP, and ATP. And yes, ATP is a building block for an RNA, okay? Okay, great. So what if it had 2 rings, this nitrogen base, it has 2 rings, and it has no amino group on the carbon number 6. Now what is it? It's a guanosine. And so guanosine, if there's 1 phosphate, it's going to be called guanosine monophosphate. Now, if it lacks an oxygen here, like in this case, it's going to be called simply deoxyguanosine monophosphate, right? And I think you get the idea by now. So if it has 2 phosphates, it's going to be guanosine diphosphate. If it has 3, it's going to be GTP. Again, guanosine diphosphate, phosphate, right? And if it has 3, it's GTP. You're going to see GTP a lot, especially in the signaling when we talk about how the cells signal or intracellular signaling mechanisms, right? So that's the AMP and the GTP. What if it's a pyrimidine? These were purines. What if it's a pyrimidine? So it could be cytosine, right? The nitrogen base could be cytosine. And in fact, if it is, so again, you could say that it's going to be then cytosine monophosphate or cytosine diphosphate, right? If it has two phosphate groups, or cytosine triphosphate. Now, of course, you would add if it's part of DNA, if there's missing oxygen here, you're going to say it's deoxycytidine monophosphate, deoxycytidine diphosphate, deoxycytidine triphosphate. If it's uracil, you're going to say uracil monophosphate, uracil diphosphate, and uracil triphosphate. Of course, if the uracil is missing an oxygen group here, it's going to be deoxyuridine monophosphate, right? Don't fall for it. Uracil is never present in DNA. Well, not normally, okay? So you're not going to see it as, you might see it at dump, but not incorporated as deoxy, but it will not be incorporated into DNA. So in fact, this form, you could see it, but it's never going to be incorporated into DNA unless there's an error or like cytosine deamination problem or something like that. Thymidine, the same basic principle. Okay, let's remove this. We kind of, we went off balance. So the backbone, let's continue. Anyway, this is a nucleotide, and now this nucleotide is really, it's ready for building. It comes and let's make an oligonucleotide, let's make polynucleotide, let's make it into a single-stranded DNA, so then it could base pair with another strand and give us a double-stranded DNA. How does that happen? Listen. So, this is an active nucleotide, right? Suppose there is another nucleotide here that's carrying another nitrogen base, right? Here's the 5' end, and it has phosphate on it. Now, it actually has to have 3 phosphate groups, but for simplicity, for now, because this is very important. Remember we said that we use phosphate for energy. Energy comes from these 2 phosphates, okay? Anyway, for simplicity's sake, for now, let's imagine that this is also, an active nucleotide, right? And here is the 5' end, and of course this is the 3' end. What's going to happen is that this nucleotide is going to come here and stack right here, right? So basically, this whole nucleotide comes and transfers over here, and of course it uses the phosphate to link the 2 nucleotides with one another, right? So a phosphate here, and here, and now here's a nitrogen base, right? And the 3', here's the 5', there's the 2', remember, and a hydroxyl group, right? So what happened here? Basically, the phosphate linked these 2 nucleotides with one another. Now, suppose you've got even another nucleotide. Same idea, same concept. So phosphate comes, right? It links another nucleotide with it, right? Here's the hydroxyl group on the 3' end, and here's the 5' end, right? Now, if you want to read this, and this is directionality, by the way, let's remove this 5' end because, it's in the middle, nobody cares about it. Let's remove this because, again, it's in the middle. And so this here, because remember, this carbon in the ribose sugar was the 3', so this would be called the 3' end. The phosphate here, remember, this is a 5' end. And so the way you would read it is, this is the directionality, 5' end to 3' end. Okay, that's all it is. Now, what do we mean when we say the sugar phosphate backbone? Look, what do we have here? It's part of the sugar, right? So the ribose sugar. So here's the sugar. What's this? This is the phosphate, right? Here's a sugar, here's a phosphate, here's a phosphate, here's a sugar. Again, here's a sugar, and here is a phosphate. And so this whole structure, now if we were like to condense it into, we've got 1, 2, 3 nucleotides, right? And 3 nucleotides, basically, This is the sugar phosphate backbone because it's made out of sugar and phosphate. That's where the weird sugar phosphate backbone comes from. Okay, what about directionality? Well, here's the directionality. 3 prime, 5 prime. 5 prime because, you know, is the place where the phosphate attaches to, and 3 prime here is on the end. If you add another nucleotide here, this would cease to become the 3 prime now, and in fact, the 3 prime end would be the end of the incoming nucleotide, right? Great, okay. So why the negative charge on the DNA? What makes the DNA have a negative charge? Well, again, it's going to be phosphate. Now, I could have sworn we had 4 points. What were the 4 points? So we talked about how the phosphate makes the backbone. Oh yeah, listen. So how can I forget such a thing? Energy. Energy, right? Listen. Okay, we need the whole board for this. Sorry, we need it, okay? Before we talk about how phosphate is going to be used as energy to make a DNA molecule, there's a couple of things we have to know. First of all, where does the phosphate come from? Okay, so let's look at the whiteboard here. What's this? This is just a nucleoside, right? Here you've got a nitrogen base, you've got a ribose sugar. Let's make it ribose, for instance, okay? In fact, we're talking about DNA. Let's keep it PJ friendly and make it deoxy. Okay, so this is a deoxy nucleoside, right? Here, NS for nucleoside. Let's change the pen, right? And so, how are we going to take okay, before that, what are these? Look at them. So basically, let's look at the sugar first of all. What do we have? We have phosphate, we have sugar, and we have a nitrogen base. Now, the nitrogen base, it's like it has 1 ring and 2 rings, and at the 6th carbon, it has an amino group. And so this means that this is an adenine, right? What are the sugar? Is it ribose sugar or deoxyribose? We look at the 2 prime. It is what? It is ribose sugar. Great. What about the phosphate? How many phosphate? 1, 2, 3. So 3 phosphate, right? So at this point, we call it adenosine triphosphate. What about the others? Well, the same thing, right? This is also an ATP, and this is also going to be an ATP. Adenosine triphosphate. And so it's a nucleotide, right? That has 3 phosphate groups. Now, this over here is a deoxynucleoside. And so it could be really anything according to what the nitrogen base is. So let's suppose that this is going to be guanosine, right? That this is a guanine, and therefore, of course, it's deoxy, because look at the 2 prime end, right? There's a missing oxygen group, so it's deoxyguanosine. So how exactly do we take this deoxyguanosine and make it into, deoxy, for instance, deoxyguanosine triphosphate, if it has 3 phosphates, or diphosphate or monophosphate? Well, you put a phosphate on it. If you put 1, it becomes monophosphate, 2 diphosphate, 3 triphosphate. Yeah, okay. Phosphates is not going to be present just freely roaming in the cell. It has to be attached to something. It has to come from somewhere. Now, since we're building a nucleotide, we're going to make a nucleoside. It comes from a source, and that source is the ATP molecule. You're going to hear a lot about the ATP molecule, people. ATP is considered like the currency or the energy currency of the cell, right? Let me tell you something before I go there. Listen, ATP is considered the energy currency of the cell because, triphosphate, because for the mere presence of 1, 2, 3 phosphates on it, right? However, there are other things, there are other currencies to the cell as well, depending on what you're building. For instance, if we want to make proteins, right? We're going to talk about this in translation later on, but let's suppose that you want to build protein. What's the energy currency going to be? It's not going to be ATP. Actually, mostly it's going to be GTP, right? guanosine triphosphate. And we know that, if you want to make a guanosine triphosphate, just remove the amino group and put an oxygen there, right? And now you have GTP. And so this becomes GTP, not ATP, and you can use it for protein synthesis, right? What else? Yeah, CTP, cytidine triphosphate. Basically, cytidine, again, remember, it's only one ring and has an amino group on it, right? What do you use? So GTP, again, for protein. Here we go. Here's protein synthesis. CTP, we use it for phospholipid synthesis. UDP, uridine diphosphate, only 2 phosphate groups, by the way, and we use this in glycolysis, okay? Or I think glycogen synthesis, glycogen synthesis, okay? Now, and there is something else. But let's leave it PJ friendly. We're not going to talk about it here. It's going to take a lot of time that we need, okay? So, the main idea I want to tell you here that yes, ATP is the main currency of the cell. However, it depends on what the cell wants to build. If it's going to make protein synthesis, it's going to use GTP. If it's going to make phospholipid synthesis, we're going to see CTP being thrown around a lot. And we're going to encounter this when we talk about biochemistry. Uridine diphosphate, when you want to make glycogen, right? Okay. So let's remove this. Let's bring back our ATP. So, 1 ring, 2 ring, and here's an amino group. Okay? Again, this is going to be an ATP molecule. Okay, nice. So, we want to make this nucleoside into a nucleotide. And suppose that this is guanine, right? Suppose this is going to be guanine, and so you want to take the deoxyguanosine and make a deoxyguanosine triphosphate. How do you do that? Well, that's simple. You take phosphates and start stacking them. So 3 phosphates, yeah, great. Where do we get the phosphates from? A phosphate, when we are building a nucleotide, is going to be coming from ATP, adenosine triphosphate, through a process called kinase phosphorylation. So, what does kinase phosphorylation exactly mean? It's very easy. It's extremely easy. Listen, phosphorylation simply means to take something and put a phosphate on it. So, listen, we have Potman here, right? I was gonna, plant something in it and, water it and stuff. And now I just use him as an ashtray and as an observer. Anyway, Potman here, if we take a phosphate on it and put it on him, it's gonna, we just phosphorylated Potman, right? So we did a phosphorylation process. Okay? Now, Potman's gonna observe us from there. Back to seat, Potman. Yeah. Okay. Good, Potman. Anyway, the cup. Let's say that we want to phosphorylate the cup, okay? Or we put a phosphate on the cup. Now it's called a phosphorylated cup. Great. What about kinase? And remember, when we talked, this is why I tell you to look at the suffix and prefix before every lecture, because everything makes so much more sense, right? What about the kinase? Kinase. So look at the suffix is, that means it's an enzyme. What does the kinase do exactly? The kinase is the hand that takes the phosphate and places it onto something. And according to the target, it's going to be called. For instance, listen, this is guanosine, right? So kinase or guanosine kinase, or in this case, deoxyguanosine kinase. There is an enzyme, and it's called, for instance, deoxyguanosine phosphate, right? Or sorry, deoxyguanosine kinase. What does it do? Well, deoxyguanosine simply refers to this. Kinase it's going to take a phosphate group. So it's going to take, by the way, okay, we'll go back there. So what's going to do is it's going to take a phosphate group and let it be from this one here, and it's going to insert it at the 5' end of the guanosine. And now we have deoxy, because remember there's no oxygen here, so it's deoxyguanosine monophosphate, right? Which enzyme did the job? The deoxyguanosine kinase. You could say the kinase. It's an easier way to remember it. A kinase is going to take a phosphate and put it on deoxyguanosine phosphate. Here. Deoxyguanosine. And so the enzyme, what's it called? It's called deoxyguanosine kinase. Okay? Great. So, at this point, the deoxyguanosine is now a nucleotide. It's called deoxyguanosine monophosphate. So it has one phosphate. Great. Now we need two more, right? My desk is too cluttered. Anyway, we need more, right? So what do? Another kinase comes and takes it from this phosphate group and inserts another phosphate right here, right? So now, what's this kinase? What's this kinase called? The one that took the phosphate from this ATP and put it right here. What's it called? It's going to be called deoxyguanosine monophosphate kinase, because it took a phosphate and put it here, so it's a kinase. A kinase to what? What's the target? The target is, well, it's going to be the deoxyguanosine monophosphate, right? So suppose now I've got my second phosphate. What about the third phosphate? The third phosphate, the same idea. We take it from here via an enzyme and we insert it here. So what do you think that, and now of course, we get, when we had two, they were called deoxyguanosine diphosphate, and when we have three, it's going to be called deoxyguanosine triphosphate, right? Now, what's the enzyme's name that took that last piece, the third phosphate, and put it right here? It's going to be called deoxyguanosine diphosphate. Why? Well, because at the time, the target only had two phosphates, and the target was deoxyguanosine diphosphate. So, a kinase, it depends on the target, right? And by the way, you're going to see the kinase enzyme a lot in biochemistry. I'm going to give you some small examples of this. Okay, listen. Let's remove the phosphate because probably should have left these phosphates. Let's just do them quickly. Okay, great. Okay, let's remove these pathways and talk about where we could find kinases. It's going to be very brief, I promise you, like 1 minute. Okay, listen. Kinases are enzymes that take phosphate and put them on a target thing, okay? So, where will you see it? In metabolism, for instance, glucokinase, right here, glucokinase. People, look at the word, it tells a lot. First of all, is for enzyme. Kinase is an enzyme that puts phosphate on something. What does it put a phosphate on? It puts the phosphate on the glucose, right? So, glucokinase. Okay, what else? Well, phosphofructokinase. Do not get afraid of the word. It seems scary, it's extremely simple. I promise you that, right? So, phosphofructokinase. This is an enzyme. Enzyme kinase, it's an enzyme that puts a phosphate on something. Where does it put the phosphate? On the phosphofructose. Okay? It takes a phosphate and puts it here. Now, where it gets the phosphate from? Usually from ATP. Could take it from GTP. It depends on the scenario, really. It depends on what you're trying to achieve, right? Because we talked about that. So if you're making protein synthesis, listen. Signal transduction, for instance, and we're going to talk about this also. Protein kinase A. So it's a kinase, which means that it takes a phosphate and puts it on a protein called A. Close enough, yes. It phosphorylates a protein. For instance, a very important one is something called the tyrosine receptors, or the tyrosine receptor kinase. So it's a kinase, so it puts a phosphate on the tyrosine receptor. And this is very important, especially when we talk about the insulin receptors, etc. Kinase simply phosphorylates something. And by the way, here's a piece of information that's quite interesting. You know those people that go to the gym and they're like man, I'm drinking my creatine, just leave me alone, I'm happy, I'm lifting weights. Creatine, by the way, or creatine, according to where we're from, is one of the most studied supplements on God's green earth. It's very safe, and in fact, is very effective. In fact, if you go to the gym, if you're trying to lift weights, trying to increase your muscle strength, the best supplement for you is going to be creatine. Why? Look how creatine works. So, creatine, and yeah, it does have something to do with kinases. So here's a dude, and he's happy. Why is he happy? Well, because he has these deformed Popeye muscles or whatever, right? Here are his muscles. And this dude is muscular because this dude takes creatine. Approximately 5 grams a day should be enough. There's a loading phase. And anyway. And so this dude, because he's drinking creatine, what's happening is that the creatine enters into the muscle cells and then gets transformed into something, right, called creatine phosphate. Okay, and that's nice. So what? So you've got creatine phosphate in your muscles. Well, listen, when we are using our muscles, when we're contracting those muscles, when we're doing, muscular activity or contractions, reps, whatever, right? You're lifting weights, you're using ATP. Now, every time you use ATP, a phosphate, because phosphate is a form of energy, is going to be released, and you end up with adenosine diphosphate, ADP, right? Di. It used to be tri, now it's di. What does phosphocreatine do? Phosphocreatine goes and says, well, not on my watch, ADP, and phosphocreatine, or phosphocreatine, right? Phosphocreatine via the enzyme called phosphocreatine kinase. Remember kinase, it donates or it accepts a kinase. By the way, the word phosphocreatine kinase here, it's kind of an exception to the rule, because this time the kinase is referring to the thing that's giving the phosphate, which is creatine. It's exception. Okay, anyway, so phosphocreatine kinase goes and gives the ADP a phosphate group, right? And gives it this phosphate, and so eventually you replenish your ATP. That's why creatine is such a good supplement. And by the way, creatine is found in beef and stuff. Of course, to get 5 grams of creatine from beef, maybe you'd have to eat 2 pounds a day of beef or something. But anyway, okay, this is how creatine works. Of course, when you have more ATP, it means you can last longer in the gym, you can probably, hit heavier weights, and eventually you become stronger, right? And by the way, we have a lecture dedicated to kinase alone. Okay, so that's nice. Let's go back really quickly and talk about how the ATP, what happens exactly, really quickly, right? Why the phosphate, what happens? So anyway, suppose now that this is adenosine. The nitrogen base is adenine, right? And at this point, what is it? It's deoxyadenine or adenosine. It's deoxyadenosine, right? Because still it's a nucleoside. So what's going to happen is a kinase called, because here's the target, it's going to be called deoxyadenosine kinase, is going to go to this ATP here and take this phosphate group and transfer it on the 5' end. Great. Now, of course, this ATP lost its, before that, very sorry, just let me tell you something. The way the phosphates are numbered, maybe it's important, maybe it's not. I'm going to do it for completeness sake. Listen, this is the first phosphate, the closest one to the 5' is called alpha. The second is beta, the third is gamma. Okay? So basically, just call them number 1, 2, 3. They could have done that by medicine and everything. We have to make everything so hard. Forget about it. Okay? So alpha, beta, gamma, or 1, 2, 3. I don't care. So, but in textbooks, they say that for instance, deoxyadenosine kinase is going to take the gamma. They could just call it the third one, right? Anyway, they take the gamma phosphate. And so they put it here. And of course, since this is the first phosphate here, we're going to call it the alpha phosphate, or phosphate number 1, right? Okay. What happens to the ATP? Well, it used to be triphosphate, but now it lost, a phosphate here. So what happens to it is no longer called ATP, it's going to be called ADP. Okay, great. So deoxyadenosine, since it has one monophosphate, now it's a nucleotide, and we call it deoxyadenosine mono, because one phosphate, right? So damn, you're going to see that. And that's not enough. We need 3, remember? And so again, this is the alpha, beta, and gamma, or 1, 2, 3. Now the deoxyadenosine monophosphate kinase comes and takes this one from here and transfers it here. And now this is called the beta phosphate, right? And so deoxyadenosine monophosphate is, because now it has 2 phosphates on it, is going to be called deoxyadenosine diphosphate, because, 2 phosphates, right? Now the last one, oh yeah, of course, the ATP, it becomes ADP, right? And the last one also, alpha, beta, gamma, they take this phosphate and we put it right here. And now, ladies and gentlemen, this becomes, of course, the enzyme that does this is called deoxyadenosine diphosphate kinase. And now we have what we call deoxyadenosine triphosphate, or the DATP, okay? And we saw this, by the way, when we talked about one of the clinical significances of adenosine. Remember, when we said that the severe combined immunodeficiency, I told you there's something called DATP, not to worry about it. I'm going to tell you what it is later. Here it is. It's an adenosine, or an adenine nitrogen base, that's attached to a deoxygenated ribose sugar that has 3 phosphate. We call it deoxyadenosine triphosphate, and accumulates. If it accumulates too much, it causes lymphocytes to die. The kid, he gets sick and failure to thrive and recurrent illnesses, etc., etc. Okay, great. Now this is all very nice, but where does the energy come in? Now, before I wipe this board clean, before I tell you exactly how the nucleotides, now this is a nucleotide triphosphate, how these nucleotides, they stick to one another, let me tell you something. Everything in life needs energy. Now, the more energy you put into something, the better it will be, the stronger it will be. For instance, let's say these are made out of these marker pens, right? They're made out of plastic. And so what I want to do is I want to, fuse them with one another. So I take a torch, or, look. I take this, right? And then I start, heating it up. I'll heat this also, okay? Eventually, they're going to start to melt, and I can fuse them with one another, right? Maybe it was, depending on the temperature, depending on the time I use, maybe, takes like 2 minutes, 3 minutes, and then I fuse them with one another. Now, it's plastic, it's fused, yeah. Then I come and I just snap it off. Now, if I left them, if I just left it like this, it probably wouldn't snap on its own, but with a little effort, I can snap it. Now, let's take something metal, for example. What do I have here that is metal? Not really. Anyway, suppose these are 2 pieces of metal, right? And metal is very hard. Metal needs to be, heated to very high degrees, right? So I use a very, very big torch at very high temperatures. Eventually the metal is going to start to melt, and now I can fuse these 2 pieces of metal with one another. I let it cool, and then I come back and I try to break it. Do you think it's going to be easy to break it? No. Why? Because I used so much energy just to fuse them. Now, DNA is the same concept. The same concept is done when we stick nucleotides on top of one another. We need energy, we need a lot of energy because we want it to stay stable. We don't want it, on the first thing that happens, any reaction that goes wrong in the cell to go and just snap the DNA in 2, right? That's why we need 3 phosphate groups. Because 2 of those 3 are going to be used to produce energy. Now, let's see how that happens exactly. It's going to be very easy. It's going to be painless. It won't hurt a bit. Don't worry about it. Okay? Now listen, here's where things get spicy, okay? Over here, we've got a growing DNA strand or a growing oligonucleotide strand, right? So for instance, this C is for cytidine. Remember, since it's a DNA strand, it's going to be the 2' end. There's no oxygen. It's deoxycitidine monophosphate, 1 phosphate, right? What's happening here? It's connected, this nucleotide is connected to this nucleotide via the phosphate of this nucleotide, right? And this nucleotide is connected to this nucleotide, again, through the phosphate of this nucleotide. Now, notice that over here, the hydroxyl group is free, right? At the 3' end, because this is 3' here, and 5' here, the phosphate is also going to be free. Now, suppose that this is a growing DNA strand, you know, synthesis is going on, and now what we want to do is we want to insert this thymidine triphosphate. Notice how it has 1, 2, 3 phosphate groups. Again, the first is going to be called alpha, the second is the beta, and the third is the gamma, right? So this is, and since it's of course going to be thymidine triphosphate, we don't have to say deoxy, because again, remember, thymine is only going to be present in DNA. Anyway, so we want to take this structure right here, the thymidine triphosphate, and we want to insert it into the growing DNA strand. So how does that happen exactly? How do we do it? Now, look here. This is going to be, so we took this, we magnified it, we put it right here, right? See the deoxyadenine monophosphate, and now I want to link these two with one another. Now, if you notice the phosphate here we just write it PPP for simplicity, okay? Actually, what's going on here is that the chemical structure of phosphate is going to have a hydroxyl group right here, right? And it's linked to the carbon of the nucleotide via an oxygen. Okay, so how do we take this and take this and we just add it to the growing nucleotide strand? So here's what's going to happen. The hydroxyl group of the phosphate is going to interact with the hydroxyl group of the 3' end, or in better terms, the 3' end here of the hydroxyl group on the 3' end is going to attack the alpha phosphate, which is this phosphate, the first phosphate, right? Now, it attacks it where? It attacks it at the hydroxyl group as well. So, what do we get here? What do we see? Basically, if we take both of them right here, you've got a hydroxyl group, which comes from the 3' end, and it's, attacking the hydroxyl group that's coming from the phosphate, right? And so, basically, what's going to happen is that this hydroxyl group is going to be thrown out. Along with it is going to be this hydrogen group, right? And so, you've got the hydroxyl group and you throw out another hydrogen with it, and now what do you have? You have water. This is H2O, okay? So we have H2O, which is water, and this is exactly why we call this type of reaction that occurred here a dehydration reaction, because we dehydrated the hydroxyl group from here, and we produced water. They may call it condensation reaction, by the way, also, right? Anyway, what's the end result? After we took this, and we have our water right now, right? And we took the hydroxyl group off this phosphate, and we removed the hydrogen. And so the 3' end of this nucleotide is only going to have an oxygen, and of course, at the same time, to drive this kind of reaction, the condensation reaction, the dehydration reaction, these 2 phosphate groups are going to be released, and we call them the PPI, or inorganic phosphate. When they're released, they're going to give a massive amount of energy, which drives the condensation reaction. The end result is that the oxygen here that's present on the 3' end is going to link to the phosphate right down here, okay? Now, of course, the inorganic phosphate is going to be broken down by a phosphatase enzyme, Remember, the suffix is an enzyme, right, for phosphate. And so we get this, okay? Now this kind, oh, by the way, the linkage here, the bond that is formed is called a phosphodiester bond. And it is a covalent bond. We call it also a covalent bond, right? Now, you're probably sitting there wondering, so what the hell does first of all, Draminors, it's called phosphodiester bond, and you'd be correct, yeah? That's when you have two of them. Yeah, but in DNA, we have two of them, right? Right. Do you know where they are? Listen, a lot of people say and throw around this like it's a slogan or something, phosphodiester bond, phosphodiester bond. Oh, the phosphodiester bond is broken. Now, when you ask them where the phosphodiester bond is, they start, you know, they look at your nose or something, right? Listen, I'm going to tell you exactly what a phosphodiester bond is, and I'm going to explain to you what a covalent bond means, okay? Do you need to know it? Absolutely, you need to know it. You have to know what covalency means. You have to know what the phosphodiester bond is, okay? Okay, and why we call it so. So first of all, let's break down the word. Let's remove bond so we can look at the word in a better manner. So what can you take from the name alone? Phospho, great. So something to do with phosphate, and you'd be correct. And this is the phosphate. What about ester? What's ester? Okay, or you know what? Let's call it a phosphodiester bond. So PDE, phosphodiester bond. Phospho because there's phosphate, di because, well, di means two. E is ester, it's a bond. So, in fact, there are going to be two phosphoester bonds. Now, if you allowed me, I will explain exactly how that happened. Let's remove these here, okay, and start from the beginning, right? Not really beginning, but you know, kind of beginning. Listen, remember when this, here's a nitrogen base, I don't care what kind of nitrogen base it is, but remember when this didn't have a phosphate on it, you know, and it was just a nucleoside, and then we added a phosphate, we said through kinase enzymes, and then we got a nucleotide. Okay, so, and remember how we said that the 5' end here, the 5' end, well, it's a carbon, right? Well, in fact, the chemical formula, the structure of the 5' end is CH2OH. Okay, so it has a hydroxyl group. And the phosphate, here, remember the phosphate has a hydroxyl group also, right? So it was carried by, and it was inserted right here, right? First of all, what does an ester mean? What's the word phosphodiester? Okay, what's an ester? An ester happens when an acid, like phosphate, reacts with an alcohol group, like hydroxyl. The bond that occurs, or the reaction that occurs, is called an ester reaction, or an ester bond, right? Now, remember we said, I told you we were gonna need to differentiate between acid and basic physiologically and biochemical terminology, or the biochemical concept. And here we use it yet again in the ester bond, okay? So an ester bond simply means that an acid, remember we say an acid, the phosphate group is an acid because it donates a proton, so we call it an acid. So an acid, like phosphate, right, interacts or reacts with an alcohol group like hydroxyl, okay? It's called ester, or an ester bond, okay? And so you would imagine that when I transferred via the kinase, okay, when I transferred this phosphate group right here, right? So that the hydroxyl group of the phosphate is going to interact with the hydroxyl group of the, well, CH2OH, right? This one here at the 5' end, and this one here are going to react with one another, and again, the hydroxyl group of the phosphate is going to go bye-bye. You get OH, and the hydrogen here is going to go also bye-bye. And so you get H2O. Remember? Because one H came from here, the other came from here, and the oxygen came from here. And this is, again, a condensation reaction. And eventually, let me just do this. What happens is, you know what? Here. Easier, right? What happens is, remember, CH2, you know what? Let's just remove the carbon, right? Let's just remove the carbon. So, you know, CH2 that was left and an oxygen, right? That is interacting with a phosphate. And this kind of bond is called a phosphoester bond, okay? This is called a phosphoester. Why ester? Of course, phospho, because you know the phosphate. Ester, because again, there is a reaction between the alcohol group and the acid, right? Bond. Okay, great. Makes a lot of sense. What about the other bond? The other bond happened when the incoming nucleotide came and basically the condensation reaction happened and it let go of the two pyrophosphates and we got another phosphoester bond. Because remember here, it was a hydroxyl group that interacted with an acid, or it was a alcohol that interacted with an acid, and so phosphoester bond. Now, since we have, look, this phosphate group here, it reacted, it made a condensation reaction right here, right? As I made obvious here, right? So it made a condensation reaction here, it made another condensation or a phosphoester bond here, it made another condensation reaction here, a phosphoester bond here also. And so we get two phosphoester bonds, we call it a phosphodiester bond. Okay? But the mic is working. Sorry about that, people. The mic, it died, okay? It doesn't have charge in it or something. Anyway, I hope you can hear me, but just one more point, What is, when I say a covalent bond, right? When I say a covalent bond, what exactly do I mean by covalent, right? It's very, very, very easy. Don't worry about it. Listen, a covalent bond. Well, let's look at the word again. I like words. They give you so much information and you don't have to use a lot of neurons, right? Listen, covalent. Co means together, right? Together. Valent comes from the word valency. Don't worry about it. This is actually a lot more of like chemistry than it is medical biochemistry. But anyway, a covalent bond is a very, oh, by the way, listen, remember when we said that the pyrophosphates are going to be released, the beta and gamma phosphates are going to go, right? And they create a massive amount of energy that makes this bond, the phosphodiester bond, the covalent bond. And that's why DNA is going to be very, very stable, right? Back to covalent bond. Listen. What does valency mean? Or what does a covalent bond mean? A covalent bond simply means that two or more molecules are sharing the same electrons. I know it's not really medical, but listen. Once you get it, you will never unget it. Once you understand this concept, you will never forget it. When you see covalent, you will know exactly what it means. Covalent means strong. It's extremely strong. Okay, so listen, let me give you an example, okay, of something that's covalent. For instance, water. Water has a very strong bond to it, right? Until you boil it and starts to evaporate, then yeah, but strong bond. So, two hydrogen, one oxygen. The oxygen, here's a hydrogen. Listen, here's a hydrogen, and here's an oxygen. Now, the oxygen has 6 electrons. It has 6 electrons, right? The hydrogen, it only has 1 electron. Now, oxygen has 6 electrons, but to become stable, it needs 8 electrons. It wants 8.
The hydrogen has one electron, this has one electron, but they want two electrons, right? Now, how do they form water, right? How does the bond occur? Of course, these bonds between the oxygen and hydrogen are called covalent bonds. Now, what happens is, the oxygen here, he goes and he says, listen dudes, how about we share our electrons, right? So I give you, Mr. Hydrogen, I share one of my electrons. Here's 1, 2, 3, 4, 5, 6. I share one of my electrons with you, and you share yours with me. And this hydrogen is like, well, you got yourself a deal, little buddy. And so he shares an electron with the oxygen. And now oxygen is half happy, right? Because he has 1, 2, 3, 4, 5, 6, 7. He has 7 electrons, he wants 8. And he shared this electron, for instance, with the hydrogen. The hydrogen now is completely happy, he has 2 electrons. And then he goes and he talks to other hydrogen. He tells him, listen, hydrogen boy. And that hydrogen's like, what do you need now, Oxy? And oxygen's like, listen, when I share, I'll give you one electron, you share with me your electron. And he's like, woohoo. And now oxygen is extremely happy. Why? Because now oxygen, because this one shared one electron, and this one shared one electron, he has 8 electrons, and he shared his one electron with this and one electron with this. He didn't lose it. They're sharing. Sharing is caring, people. Listen, 1 electron, 2 electron, 3, 4, 5, 6, 7, 8. Oxygen's happy. What about hydrogen? 1 electron, 2 electron, and this one, 1 electron, 2 electron. So they're sharing. This is called covalent bonding. Okay? And eventually you get something like water. Okay? What about phosphate? What about the phosphate and the oxygen here? Listen, here's a covalent bond, here's another covalent bond. So what's happened? The same story goes over and over again. Oxygen it has 6 electrons, it wants 8, right? So it goes to the phosphate and tells it, listen, Mr. Phosphate, it goes to the phosphate and asks it to borrow electrons. Now, I'm not sure about this, but I think the phosphate has 2 electrons to share, right? So it shares the 2 electrons with this oxygen, shares the 2 electrons with this oxygen. Of course, the oxygen is kind of, it's unfair, it's greedy. It knows that, its electronegative pull is going to be stronger than the phosphate. So we call it a polar covalent bond because basically, it's taking more than it's giving. That's all. Don't worry about polar covalency and non-polar covalency. All I want you to know is that a covalent bond is a very strong bond because of the electron sharing. Okay, so when you hear the term covalent bond, and you're going to hear it a lot, believe me, just know that it has something to do with sharing electrons with one another. If you want to memorize this, memorize it. Be my guest, why not? But you have a lot of things to memorize. Okay? And so yeah. Okay, so let's recap really quickly from the beginning. Don't worry, again, it's not going to take a lot of time. Okay? It's not going to take a lot of time. So, remember at the beginning of this, I think it's even a series by this time, this series of lecture, and we said that we have double-stranded DNA, and then we cut it down, made it one single-stranded DNA, right? Remember, we had 2 strands, then we had 1 strand of DNA, and then that 1 strand, we took like a piece of it that contained 50 nucleotides. At the time, probably some of you don't even know what a nucleotide is, okay? So this is, let's say it has like 50 nucleotides, and we called it a polynucleotide, and then we took like 15 of them, and now we called, so here it's less than 15 nucleotides, and now it's called an oligonucleotide, and then, we took 1 nucleotide, and we saw that the nucleotide was formed simply of a nitrogen base, here's NP, that's attached to a sugar, which is ribose. Here S for sugar, that's attached to phosphate, and we removed the phosphate, and this of course was called a nucleotide, and once we removed the phosphate, we simply had a nitrogen base that was attached to a sugar, and we call this nucleoside, right? And then, we just parted between the sugar and the nitrogen base, and that's that. Then we talked about the sugar, the characteristics of the sugar, why it's the clinical significance of it. Then we talked about the nitrogen bases and how to tell them apart. Then we talked about the phosphate and the reactions, and of course clinical significance of the adenosine and the guanosine and the cytidine, right? Basically, if you made it this far, you're a goddamn hero. Now, in the next lecture, we're going to be talking about DNA organization. Everything goes downhill. You already did the very hard part. From now on, if you ever get confused, I try to simplify everything as much as possible. If you ever get confused, just go back 10, 15 seconds. I don't think you're gonna need more than that, okay? So I'll see you on lecture 2, DNA organization.